So let's first discuss about cytoskeleton. As a cyto means cell, skeleton means bone, so cell bone. So it's a fibers, usually in eukaryotic cell, in the cytoplasm. However, this is sometimes very dynamic. That's a little interesting. This simple cartoon shows a spatial distribution of this fibers inside the cell. So we will discuss about three kinds of cell cytoskeleton. So the first is actin filament. You can see that usually close to this cell membrane related to, you remember the white blood cell neutrophil is chasing. They have to actively changing and their cell membrane that's involved with this actin filaments. While these microtubules having the centrosome and then it goes out, and then microtubule is very also important cytoskeleton, which has the highest compression resistance. And the other is intermediate filament. It's like cell rope inside connecting the nucleus and various organelles and cell surface. So their diameter or shapes are different. First, you look at this microtubule is a hollow pipe structure. So it has a very good compression resistance. And while microfilament or actin filament is only about 7 nanometer with its diameter with actin subunits. And intermediate filament is like a polypeptide chain, a rope of polypeptide chain making about a 10 nanometer diameter. So I brought this table which shows these filaments, 3 types. So actin filaments structure is kind of double helix shape, and there's actin monomer subunits. And its role is moving the cells and maintaining cell shape by pulling. While intermediate filament is a fibers it's like a fiber cables with about 10 nanometer. So it maintains the cell shape by resisting tension. This is pull. While on the other hand, microtubule is a hollow tube, about 25 nanometer with alpha and beta tubulin dimer. This maintains the cell shape by resisting compression because it is mechanically like this structure is very stable. However, so this one also, because of this is composing a flagella or cilia. So this is a nice electron microscope crosscut of this, and you see this circular one are actually microtubules. And an important role of microtubule is moving chromosomes during cell division. And along this microtubule, such as a neuron, you have very long fibers, axon track. You have to move organelles along with this microtubule. On the other hand, microtubule has so-called dynamic instability. It goes out and then it just disappears suddenly. So it's actually very dynamic when it's necessary. So I brought some of this microscopic view of actin filaments. You look at this, there's actin fiber bundles. And this intermediate filament, you can see this central part is a nucleus, how intermediate filament is distributed. And this also anchors the cell to another via desmosomes. And microtubule looks like this. So it's close to nucleus, but it dispersed, and it's in fact a very dynamic part of our cell. So the second one I want to discuss is a plasma membrane. And I titled as a selectively permeable back of the cell cytoplasm. Because of this cytoplasm, the name comes as a plasma membrane or cell membrane. And this is a back of the cell. And that is composed of lipid bilayer, or phospholipid bilayer. From biochemical aspects, this phospholipid is composed of hydrophilic head, which of course loves water, so it faces water inside, and while there's also hydrophobic tails. So when it comes inside aqueous environment, most of our body light, and this will form spontaneously a bilayer structure, so that hydrophilic head always facing outside of the cell or inside the cytoplasm, which is of course also composed of mostly water. So that lipid containing phosphate group is called phospholipid. And in fact, this is in fact containing a fluidic aspect. It's not like tight, like water. So it has a fluidic aspect. And there's a lot of things actually on the cell membrane. You can take a look here. There's carbohydrate. This is a luminar part. This is a cell cytosol part. And glycoprotein, which is sugar protein, and globular protein. So globular means globe shape. You know, the earth has a globe, so it's like kind of circle this shape. It's called globular. And you can see protein channels, and inside surface protein, and there's also connected to cell cytoskeleton. So in the end, in a nutshell, plasma membrane restricts movement of waters and ions in and out of the cell. Then right outside of the cell, what's outside? There's a extracellular matrix which hold the cells to form a tissue. So there's many molecules here. So this is lipid bilayer, as you can see, and this is integrin molecule which connect to this is actin filament inside, and also transmit a mechanical anchor to the outside. And outside of the cell, you can see lots of collagens and elastins, and here the fibronectin is connected. And so extracellular matrix proteins having a number of functions, such as for structural function. So that is a collagen, and also the elastic part, elasticity is provided by elastin fibers. So from cosmetic industry, you know that many companies sell collagen containing cosmetics, but you have to know that, and there are many reports that the collagen itself you attach on your skin will not be able to incorporate into your collagen of the skin. It has a good property of containing water, so it can moisturize, but that cannot pass through the barrier of our epithelial skin. So don't be, fooled by those. And another function of extracellular matrix is adhesive. So it is cross-linking extracellular matrix to collagen and glycosaminoglycans, and its binding site for adhesion receptor on the cell, which you can see it's called a fibronectin molecule. Or sometimes there are laminin molecule, which is binding site for cell attachment. So during some cell culture, and some special cells may not grow well on the protein surface of your Petri dish, and sometimes we coat the surface with laminin or fibronectin such that the cells can attach better. And inside this matrix, there are many glycosaminoglycan or GAGs protein, which is unbranched polysaccharide chains. And you remember this as a food supplement who suffers from joint pain, hoping that this glycosaminoglycan can be more abundant or reproduce your cartilage. And proteoglycan is these GAGs are covalently attached to a protein core. So look at this. These are GAGs and these are proteoglycans. So there's a number of questions. I made it over here. And so what's the most abundant protein in our body? In fact, total proteins, a quarter of proteins are composed of collagen. So collagen is abundant in our body, and the major component of basement membrane is laminin and collagen type 4. And who produced this basement membrane? In fact, you remember when we talk about epithelium epithelium always are sitting on top of the basement membrane. In fact, who made this? Epithelial cells are making this basement membrane. And why extracellular matrix is important for us to engineer? Because when we are producing in tissue engineering, artificial scaffold are often coated with fibronectin or RGD peptide. RGD is a single letter of amino acid with arginine, glycine, asparagine, and these RGDs are well recognized by integrins, so it provides a good ways of creating tissue engineering scaffold. So then the membrane contains a lot of proteins, and that's actually cellular gates with vital functions. So membrane proteins are attached to or associated with the bilayer of membrane of a cell or an organelle. And here's an example over here. There are like transporters, for example, like water or ions, and they function as an anchoring function. And some membrane proteins are receptor molecule, which gets the transduced the signal from the ligand and into the inside cytosol. And there could be some enzyme. So why is this so important? In fact, more than half of modern medical drugs are actually targeting these membrane proteins. So this is of extreme importance. And the function of membrane proteins, as an example, is transporting molecules. So that plasma membrane is in fact a well-regulated, selectively permeable gate. So glucose, even if it's small, it cannot pass through the cell directly. It needs to have a glucose transmitter. And ions such as sodium and potassium are very important ions, which also has passed through this channel, ion channels. And that ion channel sometimes need to work based on the energy from using ATP. And another is adhesion molecules, such as putting together the epithelial cell to cells or cell to the extracellular matrix. There's integrins, there's a selectins and cadherins. I'm not going to talk deep in here, but later on. And also a very important aspect is cell signaling through this receptor. So receptor binds to ligand, which is a binding molecule from the outside of the cell. And that ligand is giving a message. When it binds to receptor, something happens, such as conformational changes in receptor, and that gives signal trigger inside. So receptor bind, ligand binding, triggers signal for the cell to know what's going on from the outside environment. So that contains, you see multiple arrow, means various enzymes are involved for signal transduction from this second messenger. That will cause cellular response directly, or maybe gets into the nucleus where gene expression will change. So this, I brought a cell signaling diagram, which look very, very complicated. However, this is even not completely complete one. But to give you some just rough ideas, so this is cell membrane, there are receptors. So this RTK is called a receptor tyrosine kinase. Kinase is an enzyme which will phosphorylate another molecule. So let's say there's survival factor, insulin growth factor 1, that ligand attached to this, they will cause cell signaling to activate PI3 kinase, will activate AKT, blah, blah, blah. And this will eventually change the gene expression changes, such as growth factor, survival, or even extracellular matrix can mechanically signaling through the integrins. They will eventually make the cell proliferation or changes in cell signaling or changes in response, or sometimes even make the cell to die from this death signal. So the point of this cell signal network is assuming that this is a cancer cell, then we want to know how cancer cells are keep proliferating, how to block those. The important part is even if, let's say, we develop small molecule which targets, let's say, PI3 kinase or this MAP kinase, you can block it. There's always another aspect of signaling which allows the cell to avoid those single treatments. So that has been very difficult to develop a drug which is specific enough, but then the cells can avoid those. And so that there comes, if we know this more completely and can model this, there comes a systems biology approach that we may come up with an optimal or synergistic treatment, which is, let's say, combined together to giving a better killing of a cancer cell. So next time we'll discuss more about cell cycle and proliferation and differentiation.